हेलो। तो आज हम रेस्पिरेशन प्लान पढ़ेंगे, ठीक है। 10 मिनट का है, तो ज़्यादा डिटेल में होगा नहीं, तो ज़रा बस रिवाइज करो, रिवाइज करके याद करो, ठीक है सब। ओके। तो पहला क्या, ब्रीथिंग क्या होता है? इनहेल करना, एक्सहेल करना, कॉमन सी बात है। रेस्पिरेशन कहता है यूटिलाइजेशन ऑफ O2 टू ऑक्सीडाइज डाइजेस्ट फूड पार्टिकल इन सेल्स टू मेक ATP। ओके। ऑल सेल्स नीड एनर्जी, ATP फॉर डिफरेंट प्रोसेस, ठीक है। स्टार्च से हाइड्रोलाइज करके ग्लूकोज को ऑक्सीडाइज करके CO2H2O एंड ATP बनता है। ऑटोट्रॉफ्स कन्वर्ट लाइट एनर्जी इंटू केमिकल एनर्जी which is stored in form of CC bond of glucose, sucrose and starch. Animal depend on plant directly or indirectly. Okay, food is formed in chloroplast in eukaryotic and broken in down to release energy in cytoplasm and mitochondria. Okay, so formed in chloroplast and broken down in cytoplasm and mitochondria. In cytoplasm, glycolysis occur, and in mitochondria, Krebs cycle and ATS occur. Glycolysis can occur in both aerobic and anaerobic organism, but Krebs cycle and ATS can occur only in aerobes. A respiratory substrate, molecules which are oxidized to yield that carbohydrates are favored to respiratory substrate. Okay, those substrate we found after the oxidized metal means that is. In respiration, not all energy is released in one step due to loss. If all energy is released in one step, near about 90% of energy is lost. We cannot use it. Not all energy is released freely. Okay, then it is released in the form of couple of ATP. Okay, carbon skeleton produced during respiration is used to synthesis of other molecules of cells. Okay, do plant breathe? Of course, plant also need O2. Okay, to respire obviously they have stomata and lenticels. How plants sustain without a respiratory system? How? Because each plant part take care of its own gaseous exchange needs. Okay, like leaf take care of own, branches take care of wounds, roots take care of wounds, stem take care of wounds. Okay, plants respires at much lower rate than animals because they don't have to do any work. They have to still one place, not to do anything. So they need less energy. So they don't respire much. They have loose packaging of parenchymal cells and root stem and leaf to facilitate or to transport to nearby cells. Okay, they have, imagine the cells inhales some oxygen, some air, so they transport that air to nearby cells. Okay, other lenticels transport their cells nearby, other lenticels there, other lenticels nearby there. Okay, that is how they reach all the airs to nearby cells and all the cells. Okay, glycolysis, first step of respiration. Early atmosphere was reducing. Okay, no free oxygen, and early organisms were anaerobes. Anaerobes are two types: facultative and obligate. Facultative can survive with oxygen and without oxygen, but obligate cannot survive in presence of oxygen. Okay, glycolysis is a universal pathway. As I told earlier, it can work in both. It processing occur in both anaerobes and aerobes. It is partially oxidized to make two molecules of pyruvate. Okay, means one glucose one glucose turn into two pyruvate. Okay, in the process of glycolysis, it is called EMP pathway. Due to different scientists, three scientists made it. It has 10 steps catalyzed by different enzymes. Okay, now sucrose is the help of invertase enzyme, glucose and fructose. Fructose can further made to the fructose 6 phosphate by using hexokinase. Okay, means direct it goes to this step. Then enter into the glycolysis. Then glucose plus 6 carbon. In the next step, we add phosphate group by using ATP and hexokinase enzyme. Phosphate molecule is charged. It traps glucose inside the cell as charged molecule can't cross the cell membrane. That is why we got a phosphate. Then we convert with the help of isomerase into fructose 6 phosphate because fructose 6 phosphate is easier to make than glucose 6 phosphate. Okay. Then we use one more ATP to convert fructose 6 phosphate to fructose 1 6 bisphosphate with the help of phosphofructokinase enzyme. Then with the help of aldose enzyme, we broke the fructose 1 6 phosphate into glyceraldehyde 3 phosphate and dihydroxyacetone phosphate. Both are isomers. Then we glyceraldehyde 3 phosphate releases 2 NADP and form 1 3 this phosphoglyceraldehyde, phosphoglyceric acid, 1,3-phosphoglyceric acid. So means we add one phosphate in this, and then fructose conversion to phosphate, then broke it, and add one more two molecules, so 2 NADP. So this will add phosphoglyceric acid, then convert into 3-phosphoglyceric acid. It releases one phosphate and produce one ATP. ADP combined with phosphate and form ATP. And then this 3-phosphoglyceric acid goes to isomerase 2-phosphoglyceric acid. Okay. Then it is released H2O. Okay. Then it releases H2O from phosphophenol pyruvate. And this is the final step. Phosphopyruvate releases that phosphate. Phosphophenol pyruvate releases that phosphate and convert into 2 pyruvate, okay, and releases ATP. Now, glucose in after glycolysis from pyruvate, it is 2x. This is 2x, and it goes to lactate dehydrogenase and alcohol dehydrogenase. It goes lactate dehydrogenase. Lactate form lactate when we exercise, and NADH again go to the NAD, means only 2 ATPs form here in this process, and after pyruvate goes to the alcohol dehydrogenase or pyruvate decarboxylate, it forms ethanol and releases one CO2. Okay, in this also NADH goes to NAD plus, and it also makes 2 ATP only. Then the pyruvate is called pyruvate dehydrogenase, not decarboxylase. Dehydrogenase is from acetyl coenzyme plus releasing CO2. It converts NAD plus into NADH. Then acetyl coenzyme goes to Krebs cycle. Okay. Now Krebs cycle is an aerobic respiration. It cannot done by anaerobes. Only 7% of it energy is stored in glucose is released. Hazardous product form करता है कौन? Glycolysis. And like alcohol, lactate is produced alcohol. Yeast poison sisters 13% of alcohol. Okay, aerobic respiration leads to complete oxidation of glucose. Oxidative decarboxylation of link reaction. Okay, link reaction is formation of acetyl coenzyme. Hence, pyruvate goes to mitochondria. Okay, means mitochondria. Then pyruvate with the pyruvate dehydrogenase formed acetyl coenzyme and one NAD plus. There are two pyruvate formed. The 2x into 2x, then 2 NADP. H forms 1 NADH. I'm sorry. 1 NADH forms 3 ATP, and 1 FADH forms 2 ATP. Then we comes to Krebs cycle. In the Krebs cycle, Cricket Council of India appoints smart staff for main office. Citrate, cricket for citrate, council for cis aconidate is cricket. Cricket is for citrate, council for cis aconidate, India for isocitrate, where NADH is released. Is formed. Appoints is alpha ketoglutarate. Here also 1 NADP, NADH. H is on DPH in photosynthesis. So smart for succinyl coenzyme, which forms ATP, or it actually forms ATP, but it also called ATP. You can say it. Stoph for succinate, which release FADH2. For fumarate, mean malate, and for NADH, office for oxalate. Okay, here total number of ATP formed by complete oxidation of glucose glycolysis from 2 net ATP to NADH, which means 6 ATP. Then oxidative decarboxylation reaction formed to NADH and 6 ATP and ATS. TCA cycle forms 6 NADH means and 2 FADH and 2 ATP. Total 38 ATP is found. Okay. Okay, I'm going to tell you. Then see. Acetyl coenzyme goes here. Citrate. Cricket. Citrate for cricket. Council for. Council for what? Cis aconitate. Cis aconitate. Here is here. Cis aconitate. Council of India. Cricket Council of India. This is Cricket Council of India. Indians as a citrate. India release NADH and release CO2. Means you see A3 carbon, A3 carbon, this is 6 carbon, this is 6 carbon, and it is 5 carbon. It releases from CO2. The from the release of CO2. Keto goes to succinyl coenzyme. It also forms Cricket Council of India appoints alpha keto glutarate. It is 5 carbon, the succinyl coenzyme for carbon. It releases also CO2 and NADH for carbon. The Cricket Council of India appoints smart succinyl coenzyme. Smart stop for succinate. Succinyl coenzyme releases 1 ATP and succinate smart stop for succinate and F for FADH to release FADH to for your favorite main malate and N for NADH. Office or oxalate. This is how you see 3 carbon. Oh, cost 4 carbon. This is 2 carbon. Okay, this was simple. So electron transport system. So in this electron transport system, NADH and FADH oxidized होता है via ATS. Hydrogen electron sent to oxygen from H2O. ठीक है? Hydrogen electron sent to O2. The hydrogen contains 1 hydrogen, 1 proton, 1 electron. ठीक है? Complexes of ATS. First, NADH dehydrogenase. NADH dehydrogenase. First, NADH को ऑक्सीडाइज करना है. NADH से हमको ATP बनाना है तो NADH dehydrogenase, फिर FADH या फिर सक्सिनेट डिहाइड्रोजनेट बोल दो. फिर cytochrome B C complex. Cytochrome C is Y and cytochrome C and ARA3 complex and ATP synthase. Complex 1 has iron, 2 has FAD, very cool. FAS is complex in 1 2 3 FAS तीनों होते हैं. ठीक है, आयरन तो तीनों में ही होते हैं. तो कारण से स्टूडेंट बच्चों 3 में से होगा FAS और वाले में FNN and FAS, FEN and FNS 1 में, फिर FAD and FAS इसमें complex 3 में से FAS. Cis is present between complex 3 and 4. Complex 4 has 2 copper synthesis, एक CuA and CuB. Complex 5 is F not particle and F. Okay, 4H plus convert करते हैं 1 ATP में. Okay, got it. Now I have to see the so क्वेश्चन पूछ सकता है कि प्रोटॉन कौन से स्पेस होते हैं. तो इंटर क्या होता है, इंटर मेम्ब्रेन स्पेस में प्रोटॉन स्टोर होते हैं और मैट्रिक्स में क्या होता है, और इनर मेम्ब्रेन ऑफ माइटोकॉन्ड्रिया में ATS होता है. सो पहले क्या करेंगे, NADH को कन्वर्ट करेंगे NADP क्लास NADH डिहाइड्रोजनेस. NADH dehydrogenase पहला कॉम्प्लेक्स जो यूज़ होता है, और कॉम्प्लेक्स पहले फर्स्ट आता है, ठीक है, फिर कॉम्प्लेक्स 3 आता है, फिर कॉम्प्लेक्स 2 आता है, देन कॉम्प्लेक्स 4 एंड देन कॉम्प्लेक्स 5। क्योंकि कॉम्प्लेक्स 3 पे मतलब इसे भी इलेक्ट्रॉन पहुंचाना होगा और कॉम्प्लेक्स 2 को भी कॉम्प्लेक्स 3 पे इलेक्ट्रॉन पहुंचाना होगा। सो हियर इज साइटोक्रोम C विच इज इन बिटवीन साइटोक्रोम 3 एंड 4। दिस हियर यहाँ से NADH टूटता है और एक इलेक्ट्रॉन निकलता है, यहाँ से FMN जो है क्या इलेक्ट्रॉन टूट गए, तो FMN जो यहाँ से इलेक्ट्रॉन को ट्रांसमिट करता है FES को, इधर से ट्रांसफर करके यहाँ पे साइटोक्रोम B, साइटोक्रोम C1, दिस इज कॉम्प्लेक्स 3, साइटोक्रोम B C1, then here electron is with the help of cytochrome C goes to complex 4. Here succinate convert into fumarate, FAD and FES take electron to the complex 3, and complex 3 takes electron here and release proton in the intermembranous space of here, and this electron release proton here. Okay, all the proton stored here inner membrane of mitochondria होता है. So all cytochrome C goes to complex 4. Then the complex 4 using that electron to form H2O. Here is the O2 is final acceptor of electron and form H2O. That O2 use matrix hydrogen for formation of H2O, and further it releases the electrons H plus hydrogen H plus there in the intermembrane space, and then all hydrogens come to the F naught and ATP F1 ATP synthase from adios phosphate group and form ATP 4 H plus forms 1 ATP. Electrochemical carrier, ठीक है. Okay, I thought I completed 10-30 minutes. Okay, what is chemosmotic hypothesis? So 4 electron goes to F naught ATP synthase, add phosphate group and form ATP. That's all. F naught is in membrane, within membrane act as proton channels. Here 4H plus passing through F naught particles and generate 1 ATP. The more is here, the more ATP synthesized. Oxygen is not vertical. Inner membrane is enfolded, surface energy increases, F naught particle increases, and ATP synthesis increases. Respiratory balance sheet. All the jobs there are early reactions. This is wrong. Subordinate में तो नहीं होंगे. And reaction will go for oxidative phosphorylation. जरूरी नहीं है oxidative phosphorylation में है. None of intermediates is used to synthesize other molecules. ना. Only glucose is respired, not other substrate. That is not correct. Glucose is easier to study than we use glucose. Okay. Take amphibolic pathway. In the amphibolic pathway, carbohydrates, fats, and protein. Carbohydrates further to simple sugars into glucose. Glucose goes to glucose 6-phosphate, glucose fructose 6-phosphate, hexokinase, isomerase, then glucofructose. Here enzymes, and glucose 1,5-phosphate goes DAP and GDP. So it goes to the pyruvic acid formation, which forms alkyl coenzymes, goes to Krebs cycle, citrate forms fatty acid and fatty cholesterol. Learn only this. Oxaloacetic acid forms amino acid and protein, and alpha keto glutarate forms amino acid and proteins. Further, proteins goes to amino acid to pyruvic acid forms, or acetyl coenzymes forms amino acid and then protein. It can go to catabolism and anabolism. Or GDP goes to anabolism and form fatty acid and glycerols, and acetyl coenzyme can goes to form fatty acid and glycerols. Okay. So it can be anabolic calculation. So please subtract reaction coefficient CO2 evolved and O2 by O2 utilized. Reaction coefficient of carbohydrate is 1, protein 0.9, fat 0.7, and organic acid is 1.